Good morning, everyone. On behalf of the department, I'd like to welcome you to Neurology Grand Rounds. We have a fascinating topic today, and it's my utmost honor to introduce our speaker, Dr. Michael Shy. Dr. Shy is professor of neurology at Carver College of Medicine in Iowa. He obtained his MD and completed his internship at Albany Medical College. He then underwent neurology residency training at Columbia University Medical Center, followed by an NINCDS Clinical Neuroscience Fellowship. Dr. Shy completed a fellowship in neuroimmunology with the Muscular Dystrophy Association of Cedar Rapids, Iowa. He is currently the director of the Neuromuscular Division, the MDA Clinic, and the Charcot-Marie-Tooth and Neurogenetics Clinics at the University of Iowa. He's widely recognized as a world expert in the field of inherited neuropathies and is currently the PI of the Inherited Neuropathy Consortium. His primary interest and goal is to involve translational research to develop therapies for patients with inherited neuropathies and related neurodegenerative disorders. His work since the 90s has particularly involved models of Charcot-Marie-Tooth disease type B and the myelin protein 0 gene. He hopes to further the use of MRI in assessing the progression of CMT and to research additional outcome measures and biomarkers that will enable quality clinical trials for patients with CMT. So without further delay, I'd like to welcome Dr. Shy and hand over the forum to him.
Okay, so thank you very much for the introduction and also for the honor of being invited to talk to you. I appreciate it very much. And as I mentioned to Srinath, I have very fond memories of New York City. My kids were born there, and once a New Yorker, always a New Yorker. So without further delay, I'm going to get into the talk here, and I'm going to begin with my disclosures. And then I'm just going to briefly sort of go over how we characterize inherited neuropathies. So these are disorders of the peripheral nervous system, as you know, and we really separate them into two major groups. Those that are caused by changes primarily in the myelin sheath, and we loosely categorize those as CMT or Charcot-Marie-Tooth disease type 1, or disorders that primarily affect the axon, and we characterize those as CMT type 2. And overall, the clinical presentations or phenotypes are similar in what they cause in terms of weakness, problems with proprioception, et cetera. But they're not all exactly the same in how they progress over time, and I'm going to go through that with you. And then, as many of you know, there's genetic heterogeneity. So the first genetic cause of CMT was actually not that long ago, at least to me, in 1991. But right now, there are over 100 different genes that can cause CMT. There's new ones identified every year. And a number of these are available in commercial panels in the United States, but not all of them. So with this slide, I'm going to try to explain why people like me have a real interest in CMT. Because these are disorders that are caused by mutations in genes that affect the peripheral nervous system. So here, these group of disorders have identifiable causes. We know why they're caused. So then it gives you an idea of trying to look into molecular mechanisms of how demyelination and axonal degeneration occur. So it's nice for understanding the biology, but it also gives you an opportunity to try to develop rational treatments that are based on the biology. And again, none of these forms have successful medical treatments now. So really, it's the beginning of an era when we can actually develop rational treatments that can help people. And I'm going to start by showing you just 3 examples as I loosen my tie here. I hope you'll bear with me. And just to show you differences in pathogenic mechanisms, but also in presentations. So the most common form of CMT is caused by a duplication on chromosome 17 that encodes a gene called peripheral myelin protein 22, or PMP22. And that's named because it's only in peripheral myelin, it's not in central myelin. And in CMT, there's not a CMT1A, there's not a mutation within the gene. There's just a duplication, an extra copy of the gene. So you're making too much PMP22, and that's enough to cause a neuropathy. So a rational way to treat this would be to figure out ways to reduce the expression of PMP22, recognizing that if you reduce it too much, you cause another form of CMT called hereditary neuropathy with a liability to pressure palsies, where you have only one copy of PMP22 rather than the normal two. And so strategies that are actively being looked at for CMT1A involve ways to reduce the expression of PMP22 by targeting the messenger RNA in the nucleus. So one way of doing this is with antisense oligonucleotides. And these are artificial sort of DNA constructs which bind to specific, they're generated to bind to specific regions on the target messenger RNA, like up here. And when you have a double-stranded form of DNA here, that brings in RNase H, which degrades it. So that reduces the amount of PMP22. Another way of doing this molecularly is to use what's called microRNAs, which bind to the 3' untranslated region of the messenger RNA. And when you affect the 3' UTR, you can sort of tamp down the expression of a gene and regulate the gene expression with that. And so these are two examples that have actually been looked at in CMT1A. And I'm just going to show you the two papers here. So this is by the Ionis group when they used antisense oligonucleotides to, as I showed you, decrease expression of PMP22. And then more recently, the group from Kleopa in Cyprus used RNA interference and microRNAs to achieve the same goal. And in both of those cases, the rodent models were essentially cured of CMT. And so I think the question is to get these ready for clinical trials in humans. So that's one approach. And I'm going to start by just show you a patient with CMT1A.
I just need them a little further back.
Sorry, I'm trying to get the sound down there. And the reason I'm showing this patient to you is he's probably about 40 here, and he's got a steppage gait. He's not going to be able to walk on his heels, and he's going to have bad proprioception. He won't be able to tandem walk. But this is a slowly progressive disorder. He would have been clumsy as a child, like the slowest runner, problems with balance. And he's at the point here where he's going to need ankle-foot orthotics or AFOs. But most people with CMT1A never end up in a wheelchair, although they lose fine movements with their hands. And as I mentioned, they need ambulation support. So it disables them, but it doesn't shorten a person's life. And again, the point I want to make here is it's very slowly progressive throughout a person's life. Now I'm going to show you another type of CMT. CMT1A was the most common form of CMT1. I'm going to show you CMT2A, which is the most common form of CMT2. And this is caused by mutations within a gene, and the gene is called mitofusin-2 or MFN2. And MFN2 is encoded in the nucleus, but it goes to mitochondria, and it enables mitochondria to fuse together, hence its name, mitofusin-2. And the energy is supplied by this GTPase domain, and these folds are called HR domains. And you can see that the mutations that cause CMT2A tend to cluster in these regions, and they prevent normal functioning of MFN2, so mitochondria can't fuse together. When they can't fuse together, they can't get loaded up onto axons, and then they can't be transported up and down axons. And one strategy for a successful treatment comes out of the group at Washington University in St. Louis, led by Gerald Dorn. And what they've done is they've developed small molecules which enable the mitochondria to fold correctly despite the mutation. And they've looked at these in rodent models, and again, there's been success. So here is just a wild type control animal. Here's the CMT2A mouse, and you can see the reduction in the number of myelinated axons. Again, this is an axonal neuropathy, CMT2. And with the treatment, you can see there's a return, and you see it also down here in the EM studies. And I'm going to show you the phenotype here. There are different mutations, but many of them present like the ones I'm going to show you now, which are much more early onset and much more severe. So let's see if I can stop the sound first. And this little guy is 7 years old. And I think you can tell already he's more severe than the last person I showed you, but he's walking, and I think you can even see he has trouble lifting his knees in addition to the steppage gait. So he's got some proximal weakness. But this progression is not slow. And I'm going to show you the same little boy at age 10, 3 years later. You can see he's going to need an electric wheelchair to get around. So the idea that all CMT is slowly progressive is false. It's not all slowly progressive. And again, it depends on the subtype in the gene. Now, so that, again, is a dominant disorder. And the most current treatment strategy is for the use of small molecules such as those I described. Now I'm going to show you a third type of CMT, and this is an autosomal recessive form. So in an autosomal recessive form, you need a copy of the mutation on each allele. And instead of getting a gain of function from the mutated protein, here you end up with not enough of the protein. So it's a loss of function. And this gentleman, you can see his phenotype here. He delayed walking. He was the slowest runner. A characteristic feature of this particular type is scoliosis. And this young man needed rod replacement at 20. And here, I don't know if you can actually easily see this, but these are his nerve conduction velocities in his ulnar nerve. And the velocity is in the 20 meters per second. It should be 50 meters per second. So this is a demyelinating form, and it's recessive, and it has 2 mutations in the SH3 TC2 gene. And this is CMT type 2C. It's the most common recessive demyelinating form of CMT. And I'll show you, actually, before I show it to you, I'm going to just show you a strategy. So here, we're talking about not enough of the protein. So the strategy here is gene replacement, gene therapy. So the group from Cleopas cleopa has looked at a mouse model of CMT4C, and they've treated this with the AAV vector intrathecally that replaces the SH3 TC2 gene. So mock is when it's a fake vector, and full is when it's the replacement gene. And here you have the thin myelination from the untreated animal or the sham treated animal. This improves with treatment. These show G ratios. A G ratio is the ratio between the axon diameter and the axon and myelin diameter. And you want it to be lower. And with the mock treatment, you can see that it's elevated compared to the replacement vector. And the axon diameter is also increased with treatment. And in a point I'm going to come back to, when there's axonal degeneration, there are biomarkers, and one of the major biomarkers in CMT is neurofilament light. And you can see in the untreated animal, this is the wild type. Here's the mutant animal. You can see that neurofilament light levels are elevated. This is at 6 weeks of age, and here's at 25 weeks of age. And when you treat the animals, you can see the neurofilament light levels drop back down. Again, so gene replacement is a successful strategy in this mouse model, and this is also being looked at for human patients. And now, as promised, I will show you the patient with CMT4C. And I think maybe you can see the residual scoliosis. And he's not as bad as a little boy I showed you, but I think you can appreciate that he's more severe than the gentleman with CMT2A I showed you. Okay, so it's exciting that we have rational strategies to treat patients with different types of CMT. But again, the speed that they progress varies, and it's important to be able to measure the normal progression of these patients, what we call the natural history, because if a treatment doesn't immediately reverse the disease, which is unlikely, we have to determine if it's actually altering the natural history to see if it's being successful. And we have to be able to do that in a relatively short period of time, because things like clinical trials are expensive, and you can't run a clinical trial for 10 years. You really need to do this in less than 2 years, for example. And so the way we've approached this is to look at different types, first of all, of clinical outcome assessments to measure progression. Some of these are composite scales. Some of these are what we call functional scales, and some of these are called patient-reported outcomes. And I'm going to show you how we've gone about doing this. So the first thing is that not all patients with these different mutations progress at exactly the same rate. So you need to have a big enough population of patients to be able to sort of get an overall measure of how the population is doing. And we've been able to do that because we have formed what's called the Inherited Neuropathy Consortium. This joins together 20 sites from around the world. And you can see our goal is to get this natural history data in addition to identify new genes. And we follow about over 7,000 patients, and these are our sites. And we've been in existence since 2009. So we've been able to accrue some of the numbers needed to get these natural history studies. And before I get into the studies, I want to just go through a quick definition here of what's called the standard response mean, which is the mean change in an outcome measure over the standard deviation. And this is just a formula which calculates this SRM. And if your SRM ends up with a number of less than 0.2, it means the responsiveness of that outcome measure is minimal, and it would take over 1,000 patients in each arm of a double-blind trial to be able to tell in 2 years if the drug was slowing progression by 50%. If this works exponentially, so as you move up from a SRM of 0.2 to 0.5 or 0.5 to 0.8, it really affects these numbers a lot. And when you get up to greater than 0.8, you could probably do a clinical trial with 100 patients in each arm, with that being the primary outcome measure. So with that in mind, we started in 2009 with an outcome measure for CMT that we call the CMT neuropathy score. And this is a score that asks about activities, patient-reported activities of daily life, what they can feel, what they need to walk, how they use their hands. Then it has components based on sensory and motor functions on the neurological exam. And then a point I forgot to make is that even on the demyelinating forms of CMT, the clinical impairment correlates more with axonal degeneration that occurs after the demyelination. It doesn't correlate very well with demyelination itself. So there's a neurophysiologic component of the CMT neuropathy score, but it focuses on the size of the evoked amplitudes in the ulnar nerve and for sensory for the radial snap. And this is going to be not much of a surprise to you, but patients don't enjoy nerve conduction velocities, and they get to be expensive when they're repeated. So we also use a variation of this where we don't use the nerve conduction data, and that's called the CMT exam score. And if I'm off in left field, please feel free to ask me questions as I go along. Now, a score to be useful has to be what you call linear, in the sense it has to be equally able to detect changes from mild up to severe patients as you move along the spectrum. And when we looked at the CMT exam score and the CMT neuropathy score, we found that it had gaps between mild and moderate and severe. It wasn't linear. So we subjected it to a mathematical model called rash analysis to make that linear, and it just weights some of the scores. And then this has been used, and this is a paper that was published in neurology in 2020, and this is looking at CMT1A, that first model I showed you, over time using the rash-based neuropathy scores. And here, this is over time in years. So we followed patients out because, again, we've been in this since 2009, up as far as 6 years out. And we can see that there's slow progression using the CMT exam score. And it breaks down differently depending upon how severely affected the patients were when we started. So if the patients were mild, like in red here, their change over 2 years would be a mean of 1.48 points and a good p-value. And the standard response mean was pretty good at 0.55. So there, if you pick that population, you could really detect significant change with a reasonable number of patients. But if you look at the severe patients, they hardly changed at all. And again, their standard response mean was 0.2. So again, you would need a huge number of patients. So again, these studies allowed us to focus really on the milder patients for potential clinical trials with CMT1A. Again, this is using the CMT exam score. Now, we also used the CMT exam score to look at a natural history of patients with CMT2A, that second group that I showed you. And this looks like a busy slide, but what it is on the bottom, there's a bunch of different mutations that can cause CMT2A. So this is not a duplication. There's different mutations within the gene, like I showed you. And these are the various mutations. And here are the numbers of patients with those mutations. So here, for example, at this arginine 94 site, we have 31 patients. And there are other sort of hot spots where there's 16 or 19 or 17 patients. And if you really focus on those patients, you'll look and see at these red bars, this is when the onset was. So most of these really occur in early infancy. These black error bars here are the spread of the phenotype of the mutation. And in the patients where we have a lot for a particular mutation, you can see that that actually is pretty tight. So as a general rule, the mutations present in a similar way for a particular mutation. And if you look at this group as a whole over years, you can see that there's a progression using that CMT exam score. And if you look at individual mutations, where there's like 24 or 13 or 18 patients here, you can see that the slope changes, but there's still linear progression with that CMT exam score over 2 years. And this is a busy slide. Mainly, it's just to show you that this is severe. So the red boxes are when we first saw the patient. The yellow triangles are when they first needed orthotics, walking aids, and the blue diamonds are wheelchairs. So a number of these patients are in wheelchairs. But for the purpose of what we're talking about now, if you look at their CMT exam score over 2 years, it's 0.55. Again, a doable number for a clinical trial. And we're going to get into the CMT pediatric scale shortly, but we've looked at that, and that's 1.06 for the standard response mean. So again, you could do a clinical trial with even fewer patients there. This is just to show you that similar studies have been done with our patients with CMT2C, with the SH3-TC2 mutations. In respect to Tyler and the other authors, this paper is under review, so I'm not going to present the data here now, but suffice to say it's showing similar results. So that you can use the CMT exam score over time if you pick patients carefully to actually power clinical trials. Now, as I mentioned, we've had the CMT Inherited Neuropathy Consortium since 2009. And over that time, we've tried to learn new ways of looking at patients. We've also had talks with organizations like the FDA, and they really pointed out to us the need to develop functional outcome measures and patient-reported outcome measures. And I'm going to talk about those a little bit now. And another point I want to make here is that even if you successfully treat a molecular cause of a disease like an inherited neuropathy, you also have to deal with the sequelae that have happened before, and in this case, the axonal degeneration. And if there's been a lot of axonal degeneration, it's going to be a big challenge to reverse that, which is another reason that getting patients when they're milder is important for clinical trials. And ultimately, it's a reason why you want to be able to do treatments in children as long as they're safe. Recognizing that, with the help of Josh Burns and Rick Finkel, we developed what was called the CMT Pediatric scale in 2012. So this is a functional scale. So for dexterity, kids have to do things with switching these pegs around, a 9-hole peg test. There's quantitative muscle strengths. There's balance activities. Kids have to do a 6-minute walk. They have to do lunge tests. And if you look at these patients over time, these are color-coded. So CMT1A is here. CMT2A is this color. And you can see that, again, these show progression over time. And I will get into that. So a 5-year-old is not going to be able to do the same thing as a 20-year-old just because of normal aging. So what needed to be done for these pediatric scales is to normalize this to age. And what Josh Burns and his colleagues in Australia did was develop what's called the 1000 Norms Project. So they identified 1,000 people in Australia from the age of 3 up to over 100. And they developed normative data for all the components of the CMT pediatric scale in that population. And so that enables us to use a calculator. So when you put the results of what a patient can do on the individual components of the CMT pediatric scale, you click on this icon and it will calculate for you z-scores, the differences from norm for the patients with respect to their age and gender group. And so we were able to use this to look at the responsiveness or the change in CMT over time. And first of all, this is just to show that, again, the score from CMT peds goes from 0 up to 42. The higher the score, the worse. This is a CMT neuropathy score I showed you before, and this just shows that severity correlates between the two. And if you look at how the CMT ped score changes over time, and these are mainly in patients with CMT 1A, you can see a change from baseline within 2 years of 12% if they were wearing assistive devices at the beginning. If they had orthopedic surgery, it was about 14%. And if they had no intervention, it was also about 14%. So you could detect change with CMT pediatric scale over time with the CMT ped scale. And because it's a functional scale and we knew that we needed it, we modified it slightly for adults. And this is called the CMT functional outcome measure. And this was just published in 2018. So we're doing studies right now looking at how that changes with time. And I'm going to come back to that point. But again, it's a functional way of looking at how patients change in adulthood based on what they're capable of doing. And it's an extension of that CMT pediatric scale. This is just to show this is a CMT functional outcome measure, just the components of it, which are very similar to the pediatric scale. And again, it correlates with the CMT exam score, as I showed you for CMT peds. And so that just left us with one group of patients that we didn't have clinical outcomes for. And these are infants, so from birth up to the age of 4. And again, ultimately, we want to be able to treat children as young as possible with these diseases, and ideally before they even develop symptoms. So we develop what's called the CMT infant scale. And this is just some of the features of it. We know what normative values are for these, and there are gross motor functions. And there are fine motor functions. And the normative value are based on what kids are supposed to do at a particular age. And again, our goal is to try to identify patients as early as possible, but in the absence of treatments, we don't want to give an unaffected child a diagnosis that can affect how they're treated growing up and can affect things like insurance or whatever. So we tried to figure out a way to do this where we wouldn't diagnose patients before they had any symptoms. And we used that in the paper talking about the CMT infant scale. And I'm showing you the early data on this. And some of it, I think I'll show you some correlation on the next slide. But the reason I wanted to show you this slide here is because of this row, the at-risk group. So that at-risk group doesn't clearly have evidence of CMT, but they have a family history, so they're at risk to have CMT. So we get their scores. We want to evaluate patients over time, and then we ultimately go back if the CMT declares itself, and we can see what the scores were before they had symptoms. This is just a picture of the infant scale. So I hope I didn't go through those functional scores too quickly. But again, the idea is to add a functional component to our natural history data. But then all of the scales up to this point have been put together by scientists or physicians, and we needed to develop patient-reported outcome measures. And so what this is it's called the CMT Health Index. And it begins with patient interviews about what they think is important for CMT. There's initial constructs created, and it's distributed to a wider population of CMT patients for their input, revised. There's interviews that are done about it, and the final instrument is created. And that's called the CMT-HI, and this is just the same thing I just told you. And again, this has been published, and this has also been published just within the last year or so. So it's undergoing longitudinal studies now because we need to know how this changes with time. And we need to do this with children as well. And so this is a paper that was published last year, and led by Sindhu Ramchandran. And this is a pediatric version of that, a pediatric quality of life outcome measure. And this just summarizes that the CMT genotype did not seem to affect the total quality of life instrument. There was physical and mental components. The gender and severity of the CMT did impact the total and the physical components of the quality of life. Females had worse quality of life scores, and more severe CMT translated to worse quality of life scores. And again, this is undergoing longitudinal studies also. And so sort of to summarize some of these clinical outcome assessments, we have scales that look at infants, children, adults, all age groups, and we've put together online calculators where all of the scores can be calculated based on the data from the 1,000 Norms Project. And all of these have been published, and the ones after 2018 are undergoing longitudinal analysis at the moment. And this is just to summarize what's going on at the moment. So there is a U01 grant led by David Herman at Rochester for CMT1A, and that's looking at the CMT FOM, the CMT Health Index, biomarkers I'm going to talk about for CMT1A, and similar studies are going on for the forms of CMT listed below, which are some of the more common forms of different types of CMT. Okay, so those are clinical outcome measures based on patients' exams. But we also like to get biological markers of the disease and disease progression. And these are known, as you know, as biomarkers. And these are important because some of them can show responsiveness to treatment that is independent of what patients tell you. Some of them are markers of pathogenesis. And some show you that a treatment is hitting a target. And again, some of them can be more sensitive measures of disease progression, which is important when you want to have clinical trials of less than a couple of years. So let's, as we talk about biomarkers, let's go back to our CMT1A, the first case I showed you, where we have a lot of the clinical data. So again, PMP22 is a tetramembrane protein. It's just expressed in compact myelin of the peripheral nervous system, and there are several rodent models of this, and I showed you some of the results from those earlier. Now, we can't just go biopsy nerves. Several nerve biopsies have been done in the past. You only have 2, so you can only do it twice, and they're invasive. So we wanted to look for another way to see if we could measure PMP22 in nerves. And what we did, with the help of John Severin, is we looked at skin biopsies, because skin has lots of different types of cells in there, but it also has myelinating Schwann cells going around some of the nerve fibers. And with John Severin's help and a technology called digital nanostring, we were able to put together a little library of molecular constructs to genes that are only expressed in myelinating Schwann cells. And we put together a custom panel of about 50 of these. And then you can put molecular biomarkers on them that are sort of different colorimetric, like you see over here. And it's sort of like barcodes at the grocery store that each one identifies a particular myelin gene. And then just with a 2mm punch biopsy, you can use this technology, and you can actually measure the amount of myelin genes that are expressed. And this has advantages over what's called PCR. So with PCR studies, there's an amplification steps involved, so that when you're looking at small changes, that can get lost when you've amplified the expression of a gene hundreds of or thousands of times. But with this, it just counts the individual numbers, so there's no amplification step. And with this is what's called a volcano blot. These are log values, and you can see at a p-value of like 10 to the 6th, you can identify elevation of PMP22. These are just 2 exons that are used to start PMP22. So you can use skin biopsies to actually identify the amount of PMP22 being expressed in these Schwann cells. And then you can look in animal models to see if, or in patients to see if that changes. And in the animal models I talked about with the ASO treatments, for example, they were able to show in paw biopsies that you could actually draw PMP22 expression. So this is from plasma, and this is just a cartoon to show that these are obviously axons with Schwann cells around them. As axons degenerate, neurofilament light gets released into the blood, as I mentioned earlier, and you can use that as a biomarker. And this is studies that came out of Mary Riley and Alex Rosser's groups in London. And you can see that compared to controls here in CMT1A and in other types of CMT, you see expression is increased. And you can use a sort of a super ELISA technique, which is these high multiplex immunoassays, and where you have antigen target binding, but then you use DNA constructs to match with each other, and you can sort of amplify the sensitivity. And doing that, we were able to show that in CMT1A, you get increases compared to controls, like Mary Riley's group had shown. But we also were able to use this in what's called an O-link screen to identify a novel protein that actually wasn't even recognized as a myelin protein, which is called TMP-RSS5. And here you can see in CMT1A, it's elevated compared to control populations. So TMP-RSS5 is a novel protein biomarker that's elevated in CMT1A. And I can't remember if I have this in these next slides, but it looks like it's specifically elevated in CMT1A. And this is just data from different libraries, showing that TMP-RSS5 is specifically expressed in peripheral nerve and in Schwann cells, and here in different cell types. You can see it's in Schwann cells. And without getting too deep into the molecular biology, peripheral nerve myelin expression is regulated by transcription factors, including SOX10. And so if you knock down SOX10 with siRNA, you actually knock down expression of TMP-RSS5, showing that this is regulated as a myelin protein. When you want to look at biomarkers, you want to see how sensitive they are and how specific they are. And so what you do are what is called these ROC plots, and where you have sensitivity on the x-axis and specificity on the y-axis. And you like to get an AUC curve of above 0.8. That's sort of the area under here. And here we can see for TMP-RSS5, it's 0.9, which is even stronger than for neurofilament light. And this is just to show what I think I mentioned earlier, that for other types of CMT, the elevation in TMP-RSS5 isn't there. It seems to be specifically for CMT1A. This is just showing in some of the other types of CMT, we're using this O-link screening also. And the one thing that seems to be common to all of them is neurofilament light, which is elevated throughout the different CMT subtypes. Finally, we talked about microRNAs as a strategy to target 3' untranslated region in CMT1A. But the body uses microRNAs to regulate gene expression also. These are secreted from cells in little vesicles, and then they go and they travel to target cells, and they can affect the expression of various genes, including myelin genes. So what we did was we looked in plasma for specific microRNAs as biomarkers. And this is just, again, to say what they are. So microRNAs are, again, what I told you, small RNAs that are regulated and secreted by cells. And when they're made in muscle, we call them myomeres. And so what I'm going to be talking a little bit about is myomeres versus other microRNAs that are expressed by Schwann cells. And what we did was we identified a number of microRNAs. In yellow here are the ones that are made in muscle. So these are myomeres. And in green here are the ones that we've identified that are elevated by Schwann cells. And it's a busy slide. I apologize for that. But the bottom line is that the microRNAs that come from muscle, which get elevated, we think, as a sign of denervation, those correlate with neurofilament light. So the positive correlations are in red here. The Schwann cell microRNAs, they correlate well with TMP-RSS5. You see the red here. But again, as with other markers of demyelination itself without axonal degeneration, they don't correlate as well with the clinical outcome measures. And this, I'm just going to show you that this is just to show that these are reproducible if you look at the over time in patients. And this is simply to show those AUC curves again, to show that these some of these microRNAs are just as good as neurofilament light or TMP-RSS5. What these microRNAs are actually doing and why they're elevated is something that's under investigation. Now, the last biomarker I'm going to show you is one that there's a lot of push to have this be a primary outcome measure for clinical trials of CMT, because it's showing it's becoming the marker that shows the most sensitivity to change in a short period. And this is MRI imaging of leg muscles. So this is work that came out of Mary Riley's group in London again. And this is just showing you a calf muscle. Here's the tibia. And here, the black is what muscle normally looks like. Now, if muscle has chronic disease or if there's been denervation, stem cells within the muscle differentiate into fat cells. And that's shown in white here. And this is called the 3-point Dixon technique. And you can see in this 35-year-old woman here, her CMT neuropathy score is 5, which is very mild. See, there's just a little bit of fat. And here's a 43-year-old man who's got a CMT score of 19, more severe, and you can see there's more fat. And in a 64-year-old with a high CMT neuropathy score, you can see the calf is sort of full of fat. And you can actually color code the individual muscles to see which muscle group has fat in it. So for example, in this patient, in the tibialis anterior, there's 3.7%, in the gastroc, 3.6%, et cetera. And you see these numbers increase for the tibialis anterior in the more severe patients as you go up. And the key thing in this is just to show that there's a correlation with the neuropathy scores. I'm going to go through that quickly. Here is what happens if you look at these patients just 1 year later. So if you look, here's our same 43-year-old that I showed you before with a score of 19. Here's his anterior tib score. If you look at a little later, it's gone from 34 to 36%. And in most, though not all muscles, you see a similar type of increase within a year. And if you look at this population of patients, and we had 20 patients or 10 patients in this, you can see that as a group, the increase is significant enough so that you get a standard response mean of over 1. So we're down in a large responsiveness, which means if this was a clinical outcome measure, and this is just in 1 year, you could use this as a primary outcome measure and do a study in less than 100 patients of each arm. And when we did a study in Iowa and London together, so we had more patients and we did similar types of analysis, we found that, again, our standard response mean was good. It was elevated over 1, but I'm going to show you this busy slide here. So 0 here is the baseline of what the patients were like when they first came in. And then what happens above and beyond that x-axis is changes after a year. And this x-axis is fat fraction. So if this is less than 10% fat fraction. So if you eliminate that box and use start with just the patients who had a fat fraction of at least 10%, you combine the London and the Iowa groups together, you now get a standard response mean of almost 2.5. I mean, you could do a clinical trial in a year with this as an outcome measure in a slowly progressive form of CMT with about 30 patients in an arm of a double-blind trial. So that's why we're excited about using this as an outcome measure in clinical trials for CMT1A. So I'm going to stop there. And before I thank the people, I just want to summarize what I've tried to show you. That the inherited neuropathies have gone just in the last 25 years from really not knowing any of the genetic causes to knowing 100 different genetic causes, and in many of them, really beginning to define the natural history studies. The biology that's going on in basic labs has really identified rational clinical treatments for many of these disorders, which are at the point that they can be used in patients, and industrial partners are actively sponsoring getting these up and running. And using a combination of clinical outcome assessments and biomarkers, we're really at a point where we can interpret whether these medications are really altering expression. So I think it's an exciting time in our field. It's an exciting time in related fields. And I think it just shows the power of these genetic neurodegenerative disorders to really develop rational treatments to help treat demyelination or axonal degeneration in rational ways, which hopefully will change people's lives. So with that, I thank you again for the invitation, and I'm happy to take questions. And if you want me to stop sharing, I'm happy to. Okay.
Thank you for this wonderful lecture, Dr. Shy. So I had a question. As an adult neurology resident, what proportion of these cases are adult onset? And how does one recognize that it is a genetic neuropathy compared to other etiologies in adult patients? So since we're talking about such advanced therapies, how do we catch these early when it's an onset in adults?
Yeah, so that's an evolving and interesting question, because initially we sort of thought that almost all CMTs really started in childhood if you asked hard enough questions, but an increasing number really began presenting in adulthood. And one thing is obviously a family history, which isn't always successful, because even in dominantly inherited neuropathies, uh, uh, sometimes they start out of the blue in a person, you know, in a pretty high percentage of cases. And in some of these adult onset ones, they don't all start at exactly the same time. So it's tricky to interpret a family history. I think recognizing that this is just sort of my perspective, that it looks like a sort of idiopathic case is a start. For example, if somebody has diabetes and a primarily sensory peripheral neuropathy, you're pretty sure that it's going to be diabetes unless it doesn't fit correctly. And then you can look for features such as if there's abnormal foot structure or things like that, then give you a clue, or if it doesn't fit a classical phenotype. In my experience, a lot of the adult onset cases are sort of fall into this idiopathic group. And some of it comes from just getting to know the literature a little bit. So as an example, there's a relatively common sensory neuropathy that presents in adulthood, which is called canvas, which was just recognized a couple of years ago. And these patients develop a sensory neuropathy, but they also get a little bit of ataxia with it. That's more than just what you would see with proprioception. But they also can have a history of vertigo. And sometimes they'll have a chronic cough. And again, so when you have a patient with just a pure sensory neuropathy, this is a potential cause of the disease. And also patients, if the phenotype is a little bit unusual, so there's a current one where patients have predominantly a motor neuropathy, and this is caused by mutations in the sorbitol dehydrogenase gene, or SORD. And again, this has just been described in the last couple of years. And I'm going to just drone on for just a second on this, because it also shows an important point about some of these rare forms of CMT. So with SORD neuropathy, it turns out that this causes a block in the pathway to make fructose. So you get elevations of sorbitol because this enzyme gets blocked. And so there's rational treatment out there, and there's already a clinical trial 2 years after it's been diagnosed. So it's worth like investigating these. And I'm not given a very clear answer to the question, because it's not straightforward. I just think if it's an idiopathic neuropathy and that you're not satisfied with it, I think it's worth pursuing genetic testing. However, this is still an evolving field, and some of the tests I talked about, like Canvas, aren't even on panels yet. So it's just, I don't have an easy answer except to say be suspicious.
Thank you.
I like Elaine's cap.
She likes my keyboard.
Okay, if there are no more questions, I think we can wrap up right now. Thank you so much, Dr. Shy. Thanks for joining us. This was really interesting.
Okay, well, I appreciate you asking me, and again, enjoy your training, everybody. Okay.
Thank you.
Okay.
Take care.